Okay, dear audience online, now that we have reached the critical mass of more than 80 participants, it is my pleasure to introduce today's lecturer for the BIH lecture, Peter Nick Robinson. And it's really a great pleasure to have him back in the Berlin Institute of Health because he's a renowned expert in the field of artificial intelligence in genomics and clinical medicine, specifically in the context of rare and complex diseases. You should know that Peter has studied both mathematics and computer sciences in Columbia University in the USA, and also medicine at the University of Pennsylvania. And since then, he has continued to be a super qualified person in both fields. In 2000, he completed his specialist training in pediatrics at the Charité in Berlin, and then he held a professorship here for medical genomics, also in association with the fellowship at the Max Planck Institute for Molecular Genetics. In 2016, he moved back to the United States to join the Jackson Lab for genomic medicine in Farmington, Connecticut. And now we are very happy to have him back here in Berlin, but he's still continuing also his activity with the Jackson Lab so that we can build a strong bridge with them. Now his key contribution among many is the development of the Human Phenotype Ontology, which is an ontology of genetically associated diseases, which allows to assign clinical manifestations of diseases to corresponding gene mutations and syndromes. And this database now contains more than 13,000 diseases with different characteristics and 156,000 comments on hereditary diseases. Various applications allow now AI-based searches and analysis of this large framework, which gives us deep insight into the variety of manifestations of human disease and is very helpful for facilitating both diagnosis and also decision support for individual patients. Now, when you ask artificial intelligence, what is the beauty in Peter's work, and ask for a good format how this can be expressed, we come up with a limerick that was created by ChatGPT, and this should be my last point of my introduction before giving the word to Peter. And it is very complex, so I want to present it here that everybody can read it. And it says, in the realm where knowledge does soar, ontologies map traits to the core. With deep phenotyping's gaze and AI's brilliant craze, precision medicine opens its door. So the door's open for you, Peter. I stopped my screen sharing, and now the screen is yours.
Thank you. Thank you for the kind introduction, Christopher. I was a little worried with a limerick because most good limericks have some obscene line or other, but I guess that was okay. So I'm going to share my screen and go to slides. So yeah, I'm more than delighted to be back in Berlin. For those of you who don't know the BIH, we have a wonderful new research building, the Rachel Hirsch building, right next to the traditional Charité skyscraper. So today I'm going to give an overview of some of the topics of our work. So I'm going to tell you what an ontology is, why it's interesting. I'm going to show you some of the algorithms that we've developed for using ontologies to help diagnostics and translational research for rare disease. I'm going to show you some of our newer work in using HPO and other tools for precision medicine. And present a few other things. I'm going to close with some plans that are new, that are enabled here at BIH, and so that's what to expect. So why do we need ontologies? Computers are really good at doing some things. So if you want to multiply millions of numbers, that's no problem. The hardware supports that directly. But computers are not able to compute over many kinds of domain knowledge out of the box. And so we have to represent the knowledge in a way that makes it computable. This is the field of knowledge representation, which together with machine learning and applications is one of the main pillars of artificial intelligence. Over the last 30 years or so, ontologies have become the most sophisticated and primary method for knowledge representation. So an ontology consists of two things. It consists of a list of concepts, we say terms. So if we had an ontology for wine, then Rioja might be a good term in that ontology. Importantly, an ontology also contains the notion of relations between different terms in an ontology. And so if we had a wine ontology, then we would say Rioja is a subclass of red wine, and it's also a subclass of Spanish wine, and so on. So those two things make up an ontology. What I'm showing you here is not just an ontology, it's a knowledge graph. So this is a combination of several different ontologies for precision medicine. The ontology that my group develops is called the Human Phenotype Ontology. Those are I'm not sure if you see my mouse on the screen, but if you do, I'm pointing to terms from the Human Phenotype Ontology. These represent signs and symptoms of disease. I'll talk more about that in a second. But the knowledge graph also contains an ontology for diseases, including Fanconi anemia and thousands more. This ontology also includes a concept of genetic subtypes. So Fanconi anemia has roughly 20 different genetic subtypes. Those genetic subtypes are connected to variants, modes of inheritance, genes. The genes are connected to ontologies such as gene ontology that describe the functions and the malfunctioning that can occur in disease and so on. So altogether, this is a knowledge graph. It's a representation of the domain of precision medicine that makes clinical data computable. The HPO, this is a project that we developed here in Berlin. And so as Professor Baum summarized, I started my career at the Charité in the pediatrics department and went to the genetics department, and soon found out that that's a very difficult endeavor. And some of my colleagues, such as Denise Horn, were brilliant, some of the best clinicians ever, I think, for recognizing the clinical pictures of rare disease. And I thought, I just can't do this. I need some help to be able to recognize the diagnosis amongst the thousands and thousands of known and still not uncharacterized rare diseases. And so the way the ontology works is that it has one term to describe each of the individual abnormalities that you can see in rare disease. That's what it was at the start. In the past 15 years, we've built this out essentially for every disease. And so a term is placed in the hierarchy according to subclass, superclass relationships. So for instance, deeply set eye is a specific type of abnormality of eye location, and that's a specific type of abnormal eye morphology, and so on. Another sort of computational tool that we have included in the ontology are computational definitions using something called OWL description logic that define the terms of the ontology with links out to other ontologies for anatomy, for cell types, for gene function, for pathology, for embryology, for biochemistry, and a few other things, such that each of the terms is now logically defined in terms of what actually goes wrong. And this links it out to literally millions of annotations in other databases. Another thing which I think sets HPO apart from many other ontologies is that we've been very lucky to be able to involve the community. And so since 2008, we've had at least 5 workshops a year with clinical groups who have helped us to build out and improve the HPO in their areas of specialty. And many of these publications are shown here. So that now the HPO has become an international community project to represent the features of human disease. So I was talking in the past few slides about ontologies as a sophisticated computational model, but another thing is just important to say, without ontologies, we could not run the ecosystem of databases that we have today. And so in the upper right-hand part of this slide, we see a picture. This is not a pipe, and that's either a bad joke or a brilliant work of art, depending on your taste. But for a computer, it's a non-starter. And when I was starting to do research in this area a long time ago, essentially all of the databases in human genetics use different words, essentially just English language phrases, to describe clinical abnormalities, which made it impossible to compare data from one database with another, and also made it difficult to even know how to collect data yourself because there was no help, there was no standardized software. And so just interoperability and standards is an important part of the HPO. Another thing that's important is just to define what we mean. And so each HPO term is not just the label that you saw on some of the other slides, but it has an English language definition. It has a list of synonyms. It has cross-refs to other terminologies for electronic healthcare records and other things. Another thing to say is that the word phenotype is very general, and for a biologist, phenotype can mean the color of a butterfly's wing, or the weight of a whale. But in a medical context, phenotype either means a disease or a specific phenotypic feature. Phenotypic feature is what we mean with the HPO. So these are signs, symptoms, lab findings, abnormalities from X-ray and other imaging studies, electrophysiology, analysis of behavior, anything that a physician might use as a part of the clinical workup. This is intended to support deep phenotyping. So for some diseases, such as, say, Down syndrome, it's possible to recognize the gestalt instinctively. But for the vast majority of rare diseases and also common diseases, it's difficult to just recognize the diagnosis. And what doctors do is to essentially search for the individual abnormalities and use that profile, that combination of abnormalities, to make the diagnosis oftentimes based on looking things up in a book or in a computer program. And so that's what HPO is designed to support. We've been very lucky that as a part of our community efforts, groups from around the world have translated the HPO into their own languages. And currently we have 10 different languages on the HPO website. 2 are Australian Aborigine languages that are not complete, so they're not shown in this picture. But you can now, groups in Europe and Asia are now using the HPO in their native language for things like text mining, data entry, and to just communicate with people for whom English is not the first language. By the way, we are currently working on a German translation. So I'm going to show you 1 or 2 applications of HPO. I think gene prioritization is an important one. And so while last time when I was in Berlin, I was with Stefan Mundlos, who's a great department head in the Department of Medical Genetics. And so he got essentially the first genome analyzer, the next generation sequencing device in Europe in 2009. We thought, great, we'll try to use this to find a new disease gene. And we got the sequence, but the machine essentially gave us an Excel file, and there was no standard, there was no software for using that. So Peter Kravitz in my group essentially had to look up each of the variants one by one. We also developed a very clever algorithm to kind of narrow things down, but the whole process took us 2 months because there was no software that started from a standard way of representing the data. Now with VCF and many other genomic standards, we could have done that work in a half an hour. So 2 months to a half an hour, that's the kind of acceleration you get from standards. And so one of the things that HPO has enabled is that it is a standard for software to use phenotype data to help analyze exomes and genomes. And so even with the best filtering of variants, you typically still wind up with a long candidate list of 20, 30, 50 candidates. And what phenotype prioritization does is to rearrange that list such that if it works well, the correct diagnosis is at the top of the list, or in any case, close to it. And so to do that, we use the HPO, but we also use a set of computational disease models. So our database has models for 13,000 diseases. A typical model that you see here, it's a rare autoimmune disease with that's associated with about 15 different phenotypic features represented by HPO terms. That's the blue table on the right. Many of these annotations also have other metadata, such as the frequency. So how many, if you had 10 patients with this disease, how many would have a skin rash? It's not always 10. In many cases, the phenotypes are very variable. And so having frequency data allows you to run certain types of algorithms to, let me show here. So these are cases that we saw in Berlin. And this is a rare disease called Wiedemann-Steiner syndrome that is characterized by about 50 different features. This had just been discovered a year before we did this project, and we did not even realize that these two children who were seen within a week of each other had the same disease. Using the kind of phenotype matching that I described briefly, we were able to achieve a fuzzy specificity weighted matching. So why is fuzzy matching important? Well, in some cases, the match is exact. So for instance, the database, that's the blue column in the middle, this is from the literature, from the HPO database, said delayed speech and language development. So that characterizes the syndrome. Both of the children had that. The database also says short toe. The girl had no abnormality of the toe. The boy had long toe. So this is not an exact match, but by using the ontology structures, we can perform a fuzzy matching to say, well, it adds a little bit to the score. Similar things can be said with intellectual disability and global developmental delay. In many cases, that's just a difference of age, and other examples. And so using this, we were able to rank the girl in second place and the boy in first place. And with a few other examinations, we could make the diagnosis. We've implemented this software in a tool called Exomizer that we first published in 2014. This essentially does a simple idea. So we see here there's a chromosome with the genes represented as blue bars. The stars above the genes here represent variants, and the darker the star, the more pathogenic, the more likely this variant is to cause disease. And so we see here that there are 2 genes with predicted pathogenic variants. Using the phenotype relevance score, similar to what I just showed you in the previous slide, we can rank the genes according to how relevant they are clinically. And in this case, the gene on the very right and the gene on the very left were found to be clinically relevant. I'm skipping some of the details, but we're also using phenotypic profiles of model organisms, including mice and fish, and other kinds of network analysis to rank the genes according to relevance. In any case, in this example, only one of the genes was both clinically relevant and had a predicted pathogenic variant. That's the one on the right that would get ranked highly. And this approach turned out to be very effective for supporting genomic diagnostics. We know of about 5 companies who are using the software. It's open source and free, and many academic groups, including the 100,000 Genomes Project, who wrote in the pilot paper that HPO and Exomizer helped in various ways with a little over half of all of the cases that were diagnosed. And similar things have been said by NIH Undiagnosed Diseases Program and others. I'm going to switch topics now and start to talk about using HPO for precision medicine. With precision medicine is a very big topic. I'm not going to even try to cover everything right now. What I want to talk about today is ways of finding subgroups of a disease group. And so we've been looking at long COVID, and the first thing we did was to review the literature on long COVID to capture all of the phenotypic features that that you can see in that disease. Essentially, we got to 287 manifestations. About 230 of them were already in the HPO. So something like myalgia or muscle pain characterizes many rare and common diseases. There were many new terms, such as, for instance, COVID-TOE, that we had to add. And for this project, we used these new HPO terms to map data from an electronic healthcare system called N3C. This is an NIH project that at the time had about 5 million COVID patients. For various reasons, it's hard to make a diagnosis of long COVID in an EHR, or it was at the time we did this project, but very recently there was a new ICD-9 code, U09.9, that we could use to pick out patients from this resource who were diagnosed with long COVID. And we used those for clustering. I'm going to mention it now, but we had 6 hospitals or data partners, the initial analysis was only done in data partner 1. So I showed you a little bit about the fuzzy specificity weighted matching. We can also use this for unsupervised machine learning to gauge the pairwise similarity between patients. And so in this example, these 2 patients, patient 1 and patient 2, shared some of the same HPO terms, such as headache and depression. And in some cases, they had slightly different phenotypic manifestations, visual hallucinations and auditory hallucinations that were still closely matched and still specific. So this was a good match. These patients were judged to be very similar. Here's another pair of patients, patient 3 and patient 4. They do not share more than 1 term in common, and many of the other terms are not related closely. So you have to go far up the hierarchy to find the common ancestor that determines specificity. So we can use this to create a pairwise matrix of patients. So here you see about 2,500 patients who have been clustered using the procedure I sketched in the previous slide. And by various means, we determined that there are 6 clusters. I'm going to go quickly over this slide, but this is a so-called upset plot that shows the combination of features in sort of the upper level HPO organ systems, such as lung, neuropsychiatric, cardiovascular, gastrointestinal. So cluster 1 was seriously affected with, and patients typically had manifestations in multiple organ systems. In cluster 2, it was there was a focus on pulmonary manifestations and so on. So the clusters 2 to 4 were relatively specific for certain organ systems. This slide is showing you the individual HPO terms that contributed to that assessment. So these are according to organ systems. This is lab, constitutional, neuropsychiatric, cardiovascular, gastrointestinal. And what we saw is that we could cluster the patients. Another thing that we saw is that when we used the cluster definitions from Hospital 1, we got extremely significant generalizability in hospitals 2 to 6, and not only and this z-score, which basically shows that we would never have gotten the same amount of matching by chance. When we look at various parameters that were not used for the clustering, such as mortality, we see that there's a good bit of agreement across the data partners. So the system that we developed showed significant generalizability. This is a picture from one of my colleagues in the USA near their house, and I think it's just a lovely picture that we managed to get onto the cover that time. So I think I'm doing okay on time. I'm going to switch gears again and talk about phenopackets. So many of us who learned clinical medicine learn to phenotype like this by examining a patient and writing down the findings with on paper. Interesting, there's also several, this is from a couple of, I think about 2 centuries ago. There are some spelling mistakes here that would have made it difficult for text mining to capture this. But I mentioned earlier that standards enable computation. And there wasn't, before we did this work, sort of a general standard for representing one case report in a way that is AI ready. Some standards that are used in EHR settings, such as OMAP and FHIR, go part of the way, but they typically require additional processing of the data to get it ready for your statistical analysis. That was our goal. And between 2014 and 2022, I was co-leading a group at the Global Alliance for Genomics and Health. We had on the order of 200 meetings with stakeholders in GA4GH and interested researchers to try to figure out what are the data fields that we need to provide a general model for computation, not just across phenotypic features, but essentially across the entire record of healthcare, for research purposes, with the goal of integrating data with genomic data. And so HPO, what I've been talking about so far, would go here in phenotypic feature, and so I'll show you each of these boxes essentially itself a hierarchical model with various fields. But in addition to phenotypic features, there are numerical measurements. There are various ways of representing treatments and other medical actions. There is a biosample for specimens, biopsies, things like that. And we're using another GA4GH standard called VRS to represent the interpretation of important variants. So this gives you an example of how this interacts with HPO terms. So previously, if people were using HPO software, they would send a list of terms. Honestly, we've gotten lists of HPO terms as Word files, as Excel files, and emails. There was no standard, so every time we wanted to do something with it, we would have to reformat. But this does more than just providing a standard format. So in this case, infantile spasms is a kind of epilepsy. With this phenotypic feature, when one can denote the degree of severity, severe, the HPO has on the order of 500 modifiers. So I studied Latin for about 8 years, so I allowed myself the joke of naming this term quotidian. And for those of you who didn't study as much Latin, this means daily. We should probably go back and change the label. And the age of onset was infantile. This is an HPO term that essentially means in the first year of life. And I wish that we could say offset to denote when something gets better, but it's either abatement or resolution in English. And so this child stopped having infantile spasms at the age of 4 years and 2 months. They were also found not to have global developmental delay. So this provides a lot more information than just the HPO terms. This is roughly what it would look like in a file. One of our goals is to start to bring a lot of these algorithms to the electronic healthcare record. Right now, I would say that HPO has become standard. It's used in the vast majority of human genetics departments to support work. It's starting to be used in electronic healthcare record systems. So it's being used in the Gosh Hospital in London, several hospitals in the Netherlands, and about 10 hospitals in the US that I know of, but it's going to be sort of put into the same files that are distributed to all hospitals very shortly. And so we've been trying to develop ways of working with HPO data in electronic healthcare systems, which is rather tricky. And so we joined something called the HL7 Vulkan Accelerator Project. For that, I would like to thank Sylvia Thun and Josef Schapers for help with the phenopacket schema. Also Dominic Salo for help on that project, and actually many others. In any case, so the phenopacket schema was published a year or two ago. We also got it into ISO as an ISO standard, not because we get any of this money ourselves, and the standard is the same as the open source one, but we're really trying to promote its use in hospital settings. Okay, so what I told you what a phenopacket is, but I didn't really tell you yet why anybody should care. I'm going to try to do that in the next few slides. So we think that the phenopacket schema can become a universal schema for reporting case reports and cohorts. We've gotten the agreement from a number of journals in human genetics to have authors submit their case report data as a phenopacket. We've agreed with ClinGen to start to work on a common repository of phenopackets, such that the groups who are now sort of annotating variants will, in some cases at least, also start to annotate specific variants with the corresponding clinical data. We've been working with National Cancer Institute to code a good part of their data using the phenopacket standard, and are going to be building this out in coming years. And so having a lot of data in the same standard means that people can come and write software that expects to get a collection of phenopackets with ontology terms, such that the amount of work you need to do to basically start analyzing the data is a lot smaller than it is now, when you're getting some Excel file with some bespoke data format. And my experience as a bioinformatician is that to date you spend about 90% of your time massaging data before you get to the interesting stuff. So we're aiming to build up an ecosystem with phenopackets. Just a few examples of why this of things that you can do. So we've been working on a software called GenoPhenoCore. And I can say that the very first research project I ever did was here at the Charité. And my mentor at the time said, well, let's collect clinical data from patients with neurofibromatosis type 1 and do a genotype-phenotype correlation. And so she said, there are the charts, and here's a computer with Microsoft Excel, now go to it. She was really inspired me in my career. I'm just making a bad joke. I don't mean to be at all disparaging, but so I started going through the charts and started entering the information into Excel. And after about a month, I had completely corrupted the Excel file, and it was just hopeless. We had to give up. And Dr. Tinchett was kind enough to sort of give me another topic. Why am I telling you this? Well, it's actually to this day really hard to do this kind of research, just because it's so hard to manage the data. And even if you have gotten your data into some Excel file, you still have to come and kind of figure out how to analyze that. And so by essentially starting with a collection of phenopackets, you can use the same software, and this will massively streamline the task. And so this software starts with a collection of phenopackets. Here's an example from a certain disease where we're comparing the effects of frame shift variants against all other variants, and show that there's a corrected p-value of association of frame shift variants with a higher frequency of intellectual disability. And this is a statistically significant finding. The software allows you to do this for specific variants, variation types, protein domains, and many other things. Another thing that you can now do, this is another algorithm that we've been developing over the last year that takes an existing algorithm called consensus clustering that's designed to figure out how many clusters exist in a data set consisting of Euclidean vectors, of numbers. And essentially the method applies some perturbations and sees how stable the clusters are. We've adapted that to sort of the pairwise semantic clustering that I showed in the long COVID project with some other bells and whistles that I'm going to skip for right now, to say, well, how many different diseases are there? And this is an important question because in genetic medicine, many genes are associated with multiple different diseases. Lamin A is perhaps the most well-known example. So different diseases associated with this gene or include progeria, cardiomyopathy, myopathy, and lipodystrophy, and a number of others. And so basically, the fact that there are different diseases is something that came out over many decades of research. But now that we've moved, now that we've moved into the genomic era, we're able to sort of quickly define new diseases and get hundreds of patients, but we're not really sure, should we be treating subgroups of these patients differently? Should we be applying precision medicine to subgroups of patients with mutations in some gene or not? And so this is an attempt to support such efforts computationally. And in this, in the picture we see here, we've basically put in 4 different diseases associated with lamin A mutations. Altogether, you see about 150 patients, and we see that we can actually capture the clustering of diseases pretty well. There's some suggestion that the 3rd disease actually might consist of 2 subclusters. This is something that a researcher could go and explore. I'm just going to mention briefly that we recently published the medical action ontology with the goal of starting to have a standardized way of representing treatments that have been attempted in rare disease, also treatment guidelines in a way to sort of be able to support clinical decision support software a little bit better. So I'm starting to get to the close. I have one more topic. On this slide, I'd just like to say that the Robinson Lab does, or we do quite a variety of different algorithms with different frameworks. What I've showed you so far has been sort of a subset of the phenotype matching topic. We've done quite a bit of work of variant interpretation, which is sort of the other side of the coin that you need to get software such as Exomizer to work very well. So I'm going to show you an example of that. We've also worked on various kinds of omics data, including ChIP-seq, RNA-seq, and splicing. That's become a really big interest. We've been working, especially in the USA, on observational statistics and EHR data and connecting EHR data to sort of open source ontologies for the kind of machine learning that I've shown you. And various kinds of machine learning, including especially graph machine learning. Had a couple of interesting projects recently that I'm not going to have time to talk about. So the splicing, I think most people in this audience know splicing. This slide is just intended to remind you that genes consist of introns and exons, and splicing removes the introns. In many cases, there are alternative ways of doing that, such that one gene can be transformed into multiple transcripts. Splicing-associated variants are either very easy to interpret. So, for instance, the 2 nearly invariant nucleotides on each side of the intron, you don't need any sophisticated machine learning to interpret them. If there's a change, it's almost surely pathogenic. But it turns out that this represents maybe about half of all splicing mutations that are out there. In addition to that, you have splicing mutations that occur in exons that sort of create a new or cryptic splice site. You have splicing mutations that affect regulatory elements in the introns or exons. And you have splicing mutations that affect other parts of the splicing signal that are sort of on the termini of the introns or at the end of the exons. And these have been hard to interpret. And now that we have genome sequencing, if you do actually apply any of the splicing prediction softwares to these variants, you're going to get hundreds of things that are suggested to affect splicing. So this is still a hard area. And of course, there have been tons of programs trying to predict the effects of splicing and variants. These range from early efforts that had essentially a very concrete model of splicing, of what happens in splicing using information content, to more recent efforts that were using various types of machine learning to essentially judge whether a certain sequence is likely to splice. However, that in clinical use, that's essentially a black box, although it works very well. And so our goal was to develop a machine learning tool that would be, at least as good as the current best of breed, but have interpretable features, including the information content. And what we came up with is something called squirrels, or super quick information content random forest learning of splice variants. So what we did was essentially to try to generate features for machine learning that would be most relevant. And so to do this, we essentially take the information content contained in either the splice site or other predicted parts and calculate the change. We compare the strength of the splice site to that of other splice sites upstream and downstream. For donor, we create a vector of about 10 bits of information. I'm not showing you all of them here. The acceptor site is a little bit more complex, and there's sort of more things to judge. For instance, one example is that you're not allowed biologically to have an AG between essentially the branch point and the intron-exon junction, and this is something that you can use for machine learning. So we did curation of splicing variants in the literature. I think we have the largest human curated data set that's around, and I think it's higher quality than those that you can get from some of the commercial databases that are out there. We used a relatively standard machine learning model, but we were very careful to apply proper validation and holdout procedures to estimate the performance. And we essentially had a performance that was just a tad better than Splice AI, but was better. Other people have done similar tests and now have included our project. Some other groups have shown that Splice AI is a tad better than squirrels. I guess it depends on the data set. But the advantage is that our program actually provides an interpretable output that we feel is more useful in a clinical context than the kind of black box output of Splice AI. This includes not only a nice graphic, so we actually created a new logo for showing the effects of splicing mutations that we think is better than the previous ones, but it also shows you the predicted effect of whatever mutation on the various sort of parts of the splicing project. And so these are things that would generally be comprehensible to a human geneticist. And there's one overall prediction. So in this case, it's saying we're predicting a cryptic splice site that would remove 31 bases from the coding sequence. So that's squirrels. So I'd like to finish up. I hope I think I'm on time. I can't say how delighted I am to be back at the Charité and to be at BIH. It's just been wonderful so far. I'd like to thank the entire everybody I've met at BIH. Has just been wonderful. My work has been about team science. So everything I've shown you has been not only my team here in Berlin, but working closely with about 5 groups in the US and the UK, Australia a little bit. What we're hoping to do in coming years is obviously to continue our work on rare disease. We're going to be working with at least 3 no, at least 4 European reference networks to build out specific areas of the HPO, to start to use phenopackets as a way of sort of capturing clinical trajectories, and using that data for analysis. We're planning to build out algorithms to use HPO and semantic and machine learning algorithms to look for clusters of common disease. So one area that I've become particularly interested in is mental health, and we're working with Melvin McInnes, who's leading an international group in for bipolar genomics, and we've been having workshops at NAMH and next to some psychiatric congresses to build out the way that the HPO is representing behavioral phenotypes. Very interested in improved EHR integration. I'm hoping to work with the groups here at BIH, including Sylvia Thun and others. Another exciting thing is that we're now working with manufacturers of sonography devices to essentially use the measurements to export phenopackets natively, to essentially avoid the step that somebody has to sort of extract data from a PDF file and put it into a research database. I think this is something that could be done much more in general. We're working on a number of algorithms for decision support that will integrate greater parts of our software stack. So we'll be using the Maxo ontology in ways that I'm happy to discuss if anybody wants to buy me a coffee or a beer. And we're going to be continuing to look at multi-omic data. And so Team Science, I'd be very happy if any of this is of interest to discuss collaborations within or without the BIH. So I can't say how honored I was to receive the Humboldt Professorship. It's really hard to describe. When I got news of this, I was in England with colleagues who had also become friends over the last 10 years, and I told them this in a state of shock at about 3 p.m. And we met a few hours later in the bar, and my friends had just been walking past a bookshop and they saw this book. And we met in one of these pubs in Cambridge that is both, a normal pub, but also unspeakably wonderful, life-changingly great to have some beers. So it was a really happy event. I was just completely honored. So obviously we are looking to hire people. If you know somebody who is really good at bioinformatics or related fields, please send them to me. And this slide is just to thank the many people who I've had the privilege and the pleasure to share some of my work with over the past years. So thanks very much for listening.
Yeah, thank you, Peter. Great lecture, highly relevant work. It's really amazing how this evolves to create really measurable impact for patients and communities. That's also the purpose of the Berlin Institute of Health. So that's a perfect fit to have you back here in Berlin on board now of the BIH in close connection with Charité and many other players here in the ecosystem. So we hope we can create a good niche for you to continue this exciting work. So we have already a number of questions in the chat, and if you have more questions, please type them into the chat. I hope we'll have sufficient time to answer them. So the first question is whether your work has led to the revision or refinement of disease definitions, for instance, in the case of rare syndromes, and whether there's interest in attempting to use this tool and your tools in this way.
So, well, I had a very small part in the ICD-11 in trying to define sort of multi-malformative diseases. My work is done as a part of the Monarch Initiative, and we're developing an ontology called Mondo, which is a disease ontology that does attempt to do this. What Mondo does very well is to link the, roughly speaking, 30 different disease ontologies that are out there to try to basically say this is the same disease. I'm not sure if that was your question. I think that the interesting part of this is probably less sort of the, defining disease ontologies, but it's actually trying to define, subtypes of disease such as long COVID. I think this is something that's still extremely challenging in the community. We very much intend to play a part in that. I think that's going to be a process that's going to accompany us for coming decades at least.
Yeah, thank you. The next one is extending now this to the question, what needs to be done to link other omics levels, metabolomics, proteomics, spatial omics, to the HPO terms? So in your opinion, what would be the potential added value for patient subgrouping? And I would like to extend this because this comes from Oliver from imaging unit. In this case, imaging mass spec. This can also be extended, of course, to clinical imaging data.
Hi, Oliver. Great to see you again. So there are many ways of doing this algorithmically. One is to sort of try to find clusters with HPO terms and see if the clusters have significantly different, let's say, concentrations of metabolites or gene expression or whatever it may be. The other is to cluster the genomics data and then see if they have differences in clinical data. The third way is to essentially try to do the clustering together. So, essentially, I think it's absolutely necessary to include all of the data in analyses that try to find sort of precision medicine subtypes, because it's extremely likely if you have a precision medicine subtype that you'll not only have significant differences in some of the phenotypic features, but you will also have substantial differences in omics features. This data is so noisy. It's been, sort of naive attempts to sort of cluster RNA-seq have not really helped a lot with most diseases. I think the challenge for the coming years is to sort of figure out what molecular investigations need to be done, what tissues need to be investigated. But I think the contribution of the bioinformatics that I've shown here is to essentially provide a way that will allow us to cluster clinical data with as much sophistication as has been possible for a lot of the molecular data like single cell and many other things.
Thank you. Next question refers to automated translation of lab values to HPO terms. So how did you classify values in general, or your link to HPO where no clear normal values are noted in the HPO term, like hypomagnesemia,
So,
so forth?
just briefly, LOINC is a system that describes lab tests. So for instance, a LOINC term will be blood potassium test. If you're testing any metabolite, any analyte like potassium, there are actually 10 different LOINC terms that describe the different ways of measuring potassium. And so the LOINC to HPO project essentially took data that was represented using I2B2, which is roughly speaking similar to FHIR, that actually has not only the specific value, so for potassium, let's say it was 2.9 milligrams per deciliter, but it also has the normal range, which is, I think, 3.5 to 5. And so if you know that, you can say, well, that's a low value, that's hypokalemia. Hypokalemia is an HPO term. So that's roughly how that software worked. This obviously simplifies very complex data, but it also integrates many different types of data. Our test use case with LOINC to HPO, we were looking at a data set of about 15,000 patients with asthma and other respiratory diseases. And using a very simple statistical test, a chi-square test, basically, we were able to rediscover 5 published biomarkers for asthma. And our procedure did not propose any biomarker that was not supported by publications. So that means that even though it is a data simplification, doing this type of analysis does enable some types of powerful downstream analysis. Yeah.
Yeah, thanks also for this answer. And there's another interesting question related to the junk DNA, so to say, how this relates to phenotypes, so the big non-coding stretches of human DNA, that's actually the largest part of the genome. So how can this be related
So
better
it
to
remains
human?
I have about 3 minutes, so I'm going to be a little superficial and simplify, but it remains a mystery why, the diagnostic rates for genome sequencing have plateaued at about 35%. 10 years ago, I think many people were expecting to find many more pathogenic mutations in the non-coding space. And that has not been the case to date. One of our algorithms has a machine learning score for predicting non-coding variants, and I was hoping that this would lead to finding many things, but it does seem to be the case that not all parts of the non-coding genome are as susceptible as others to variation. This probably depends to some extent on the redundancy of the enhancer system. So for many sort of gene activities, they're promoted by, multiple enhancers. And if you have a mutation in one of them, it's more like that will cause some fine tuning, but it won't be a big enough change to cause a Mendelian disease. And those enhancers that are associated with disease um, are few and far between. These are enhancers that are essentially in deserts where there's really only one, for whatever reason, one enhancer that's responsible for some developmental stages. That's easy to understand why in promoter mutations, again, there's a subset of genes that appear to be susceptible to promoter mutations because you see, multiple published mutations, let's say in some of the coagulation factors. I don't think anybody really understands this. I think it's a fascinating question to look into. My best guess is that we'll continue to incrementally increase the discovery of non-coding variants, but I think it's unlikely that they'll be as common as coding variants in Mendelian disease.
Thank you, Peter. And maybe we have time for one last question, which is a very practical question. There are a number of more questions in the chat that we will send out to you to be answered, and also invitations to beers, coffees, and meetings. So the next question that we can still take is here of the one of Max Zau regarding how can this work be integrated in the clinical electronic health record system at Charité.
I'm glad I only have 1 minute to answer this, but um, I, I I would like to see us use standards such as FHIR to develop applications that will interact with the Charité system, will be able to extract data that's within the firewall, that's for the treating physician, to organize the information, support decisions, there are technologies such as Smart and FHIR that would enable this. I think what BIH and Charité needs to do is to ensure that the new systems that are coming will support FHIR and will support this kind of interaction with translational research to slowly just improve routine healthcare.